Alright everyone, welcome to Scicomm Monday. Sorry for the little bit of audio mix-up that we had. It's always fun when you're working with new technologies. There's always going to be a few bugs here and there about stuff. So hopefully we can get this all worked out and nice and smooth. But thank you all for sticking around and dealing with some of the bugs that we have here. So for those of you who don't know, I am your host, Nicole Wood. You can either tweet me at WildlifeBioGal or at the Scicomm Monday handle. Either one works fine. And we like to make Scicomm Monday as engaging as possible. So feel free to send in your questions via the chat module on the Periscope. And then also to feel like if you have questions that we aren't able to get to, make sure that you feel comfortable with tweeting us afterwards, and we'll always be fine with getting to your questions on Twitter after. So today's guest is Lachlan Fetterplace. He is from the University of Wollongong down in Australia, but he's currently in Sweden of all places. So with that, I would like to welcome Lachlan to Scicomm Monday broadcast. Thank you for being here.
Hi Nicole, how are you? Thanks for having me.
Good. So thank you for sticking around with the fun difficulties of Scicomm Monday, but hopefully we all get it worked out and everyone can hear us just fine. So why don't you tell us a little bit about yourself, your background with your academic career, and how you got into Scicomm and kind of what research you're doing.
Okay, no worries. Well, as you said, I'm Australian, so I'm based in Australia usually, although I'm on a mountain in Sweden at the moment, but that's kind of unusual for me. And I'm on the east coast of Australia at Wollongong University, and my PhD, which I'm doing at the moment, is in collaboration with fisheries in the state that I'm in New South Wales. And I'm really interested in fish. I have been for a long, long time, since I can remember, really. I grew up inland, but used to walk down to the creek and collect fish and do all the naturalist things, really. I didn't realise that's what I was doing at the time, but I was really into it. And that eventually led me into doing a science degree, and I took a whole lot of time off after doing a few years of that and came back when I was about 29, and then that's when I really knew that I was into ecology, basically. And did a marine science degree, and then went on to do some honours, and that's when I started working with the fisheries, so Department of Primary Industries, really, in New South Wales, and did an honours thesis and started to do some research on fish, and decided I loved it, and went back and did a PhD, which is what I'm doing now. So I'm about 4 years into a PhD. So too long into a PhD, but I'm getting there. I'm getting close. Hopefully in the last... I don't want to put a date on it, but the last couple of months, so we'll see how we go there. And basically all of my research focuses on fish communities that are found on soft sediments, which is probably not as exciting as coral reefs, or people think it's not as exciting as coral reefs or temperate reefs or estuaries, but it's actually the dominant habitat type on the planet. So people don't often think about that, but the soft sediments in the ocean dominate the world, but we often don't look at them. And if you narrow it in a little bit, I focus on sand. So up on the shelf, I look at communities on sand. and sounds like it wouldn't be that exciting, but when you go and have a look, you start to see some really interesting things. And why I think that's exciting or interesting is we don't pay much attention to it, but a lot of our fishing effort happens on there, on that habitat. And when you look at things like marine protected areas, a lot of the area that's covered is actually over these sand areas or soft sediments, but we don't do a lot of research on them, so we don't know exactly how that type of management works, or if it can, or if it is working at the moment. So there's not a lot of research in that area. And so my collaborators and me, or my supervisors and I, so one from Wollongong Uni and one from fisheries, wanted to look at this issue, and so we decided, okay, we want to see what's there firstly, and so we use these baited remote underwater cameras that you mentioned, and basically that's just throwing a camera down off a boat, down to the seafloor, remotely films fish, and you throw out hundreds and hundreds of these cameras, and basically we put them in areas that are commercially fished, areas that are recreationally fished, and then areas that there's no fishing allowed, and we start to compare the communities on sand and see if they're different, or if they're not different, why are they not different, if there's different management happening there. And so yeah, that's a big part of my PhD and what I've been doing over the last probably 4 years. I think I've got over 400 of those videos, which all go between a half an hour and an hour, and they're taken in some from 10 meters up to about 60 meters depth, so fairly deep as well. So quite involved getting those really deep broths. You see really interesting things. You see things like saw sharks and all sorts of things, gurnards and all these different species that people that live on the coast, just off the coast, a couple of k's, there's these really interesting fish doing all these crazy things, and people don't really get to see that. It's usually too deep to scuba dive there, or there's no reef, so people don't scuba dive there.
Do you think that people just are assuming because there's no reef there that there isn't as abundance of fish there, and so they're thinking, oh, it's not worth our time because we're not really going to see fish, where they might actually see a whole bunch of them, but maybe a different diversity, so they might be missing out on some really cool fish that they wouldn't see on the coral reefs?
Yeah, well, from a tourism perspective, you can understand why people would go to a reef. It's interesting, and you're guaranteed to see something interesting, and it's in shallow water, so it's safe. But from like a fisheries perspective, we've always known that there's a lot out there. It's a resource that's getting used. And then when it comes back to the research side of things, at a PhD level, it's a bit of a risk to go out and do the research in that area. Yeah, it's a bit of a hit and miss, or if you go closer to shore on reef or somewhere that's had a little bit of an understanding of the system, it's more of a guaranteed result, really. Maybe that's it.
So
maybe that's it. Maybe it's just me that thinks it's interesting, maybe. So.
Oh, okay. Yeah, like.
But
I
particularly
wonder, like,
from a.
Oh, go ahead.
Oh yeah, sorry.
No.
Particularly from a marine protected area perspective, so looking at the soft sediments in marine protected areas, that's the real area that hasn't had a lot of work done it. So there's lots of fisheries work with like trawl surveys and things like that, but you can't often do that in a marine protected area. So that's why these camera systems are really good too. So you can send them down. You're not actually extracting any fish. You're not taking any fish out of the water, but you can go into what can be sensitive areas and put out cameras and sample the fish communities.
So it's
and show people what you're seeing.
Then,
yeah, a little less invasive. So that's why that technology is pretty handy.
Yeah, like I wonder with the, like you said, the kind of hit or miss thing, are you like having concerns of where you just aren't going to see any fish at all there, and then thus your data is just going to be a whole bunch of zeros, and that's kind of the worrisome aspect of it for your PhD?
Yeah, there was some aspect of that, and I mean, it's probably half of my PhD is based on BRUV methodology. So there was the risk that would happen, but that's still something interesting. If you're going into a lot of these areas off the coast and you're not finding any fish, you can still do something with that. It's just probably harder to write up, as you said, lots of zeros in the data set. But that's not really happening, luckily. So it doesn't seem to be happening.
Yeah.
We're just one working.
Yeah, for me, it's like I always feel like you might be getting zeros, but that's still telling you something. If the fish aren't there, then you have to try to figure out why aren't they there.
And if you're sampling, say, 60 locations and you're only getting some species in one or two of them, that the data set is really hard to work with, but it's still telling you that something's going on. You have these areas of either high diversity or high abundance, and then you want to go and look into why that area is particularly special. Is it something to do with the management? Say, no take marine reserve might be on that area, or is it something to do with the underlying habitat, or was it always like that? Is it irrespective of what the management is? So lots of interesting questions about
Right.
how the management acts and whether it's the management.
How long have the baited underwater or baited remote underwater videos been taking place? Is that a newer thing that's happening? Because I'm trying to think of, we have the camera traps that we put out in woods and everywhere else around here to see, land mammals and, other species. And so it's just like that seems to be, on the newer edge of technology of, people like really hardcore using that all the time now. Is the underwater camera systems, would you say that it's pretty much about the same lifespan as what we have here on land, or is that still kind of newer just because of the technical issues of going underwater?
Yeah, I think so. I think there's starting to be more and more and more baited remote underwater videos, but it's relatively new, but it's always got that time lag behind the terrestrial side of things. So probably once the camera traps were happening, it's probably 5 to 10 years before it really starts taking off underwater, just because of the technical side of things. But a really interesting thing that we're using now, though, and probably the last 5 years it's becoming really common, is stereo video. So you send down these stereo video systems, and then you use another program to calibrate them, and you can like triangulate a position in 3D space. So you can measure fish. It's really quite accurate. So you can send your cameras down. You don't just count how many fish are there and what species there. Then you can actually measure them. So that's really handy too. So kind of that little extra bit of technology is kind of on the forefront. So over the last 5 years, and that stuff's really handy.
Yeah, so. I read a paper probably 3 or 4 years ago about, I believe it was elephant seals, and they were trying to get some 3D imagery of the elephant seals to track the mass that they were losing as they were lactating throughout the season. So you're getting to do a little bit of something like that just underwater.
Yeah. So very interesting stuff.
Nice. So we've got some of your videos up here of things, so I figure we might as well share with everyone. And just a reminder to everyone out there on Periscope, if you have any questions for Lachlan, please feel free to send them in and we'll get them asked, because we know that everyone likes to ask all those questions. So I think like what
you might have to tell me what one's up.
Yeah, so I've got a picture up here of you with the blue-spotted flathead, which seems to be the main basis of your work, is this particular species. So what can you actually tell us about it? Like, best guess I could see is it kind of looked like a flounder. Is that a pretty good assessment, or?
Kind of. So they're not quite as flat as a flounder. They're a really popular angling fish on the east coast of Australia. So the genus that they're in, there's actually a whole lot of species, and they're targeted quite heavily by commercial and recreational fishers. But that one, the blue-spotted flathead, I guess it's a little bit like a flounder, but a little bit more aggressive and a bit bigger. So they're supposed to be ambush predators. They camouflage on the bottom and ambush their prey. Although some of the tracking work that we might talk about in a little bit shows that they don't always ambush, so they move around. They're quite active on the seafloor. Some of the video shows that as well. They move around. They'll come in and attack a bait and move around a couple of meters off the seafloor. But the reason that's one of the focus species is they're one of the most common species in the community. So you see them pretty much everywhere you put down the cameras. And yeah, particularly recreational fishers are really interested in that species.
Yeah, so I've got a video up of the baited remote underwater system here showing the blue-spotted flathead, and so we can kind of see them moving around. And you're right, they're not quite as flat as a flounder. They do have a little bit more verticalness to their structure, but they definitely are they pretty much always on the sediment, or do you see them? Will they go up into the water column at all?
Yeah, they'll only sit on the sediment they'll sit on the sediment unless something happens. So if a bait fish moved even up to 3 or 4 or 5 meters off the bottom, they might go up and try and take the bait fish. But they will come in. So you see them when you catch them as well, and you put them in a container. They change colour to try and match the container. So they have this. They can change their colour to suit the substrate, and they can be quite white, or they can be really, really dark as well. And they change their spots, which is really interesting. So you have them. If you have them on a tank, you can almost put a white bottom and a black bottom in the bottom of the tank, and they'll swim and they'll change colours. So that's pretty interesting too. So they're obviously doing something with the camouflage, whether it's defensive or to help them prey on something. I don't know at this stage.
So is it like a pretty instantaneous color change, or is it a more gradual color change?
More of a gradual thing. So it's not like a chameleon on land or something, but maybe like over 5 minutes they can change color completely.
Oh, that's pretty impressive. So
yeah,
I can't change color that fast unless I'm blushing really badly. So.
I get sunburnt pretty easily, so maybe I could do that.
So I've got the picture up here because you mentioned about tracking them. So can you tell us a little bit about how you track them and where you're tracking them to, a little bit more about that aspect of it?
All right. Well, so when I talked about using the baited remote underwater video, that's really to look at so you're looking at abundance metrics and what species are there. And then some of the results we're starting to see, for some of the species, you might not have seen a very big difference across areas that there was no fishing happening and where there was quite heavy fishing happening. We weren't seeing big differences in the numbers of fish, and so we start to think about why that's happening. And we didn't really have any information on how any of the fish in this community move. So if the fish are in an area where there's no fishing, but they often move into areas where there's commercial fishing or really heavy fishing, then they're not really in a no-take area. They're also being fished at some stage in their life. So you're not likely to see a big difference in the populations. So we were interested in finding out how they move, and decided that the next step in the project was to start to track some of the fish. And I don't know if you know much about the tracking side of things underwater, but we use acoustic trackers.
Okay.
So it's sound-based, and so we put these little tags in, and they basically send out an acoustic signal telling you where the fish is, and it's like a unique code that says "I'm here" over and over again, and you set it to whatever timing you want it, because the problem is that you can't use the same systems as you do on land, because those signals don't travel through water. So you can't send GPS signal through the water. You can't send Wi-Fi or very easily. And so you're relying on something like acoustic tags, which send out a sound signal, or satellite tags where they need to breach the surface. So you often see big sharks that are tagged on the fin. Mm-hmm. The tag needs to come above the surface. If you have something that doesn't come up to the surface, you need a sat tag that will come off the shark and come to the surface and send the information. So that it's a little bit more difficult than on land. But anyway, so we have these acoustic tags. We go out and tag. The first species that we tagged was the blue-spotted flathead, and working with fisheries in Jervis Bay Marine Park, so a big marine park on the southeast coast of Australia. And to pick up the signals, you need these tracking receivers. So you have a whole load of those out in the water. I think in that marine park we have about 50 at the moment. And if a fish swims within a couple of 100 meters of that, then you get a presence absence where the fish is. So it's starting to work out how the fish are moving between zones in the marine park. So there's some areas that are no fishing, some areas that are only recreational fishing, and then outside the park there's all sorts of fishing. And so we're starting to get an idea of how they move between those areas. And also within that tracking array, so we call it a tracking array, we've got this one small area which is called this VPS system, and that's made by a company called Vamco, and lets you work out not just where the fish is, a presence absence, to within hundreds of meters. It lets you know where the fish is to within meters, and you get this really fine scale position of the fish, almost like a GPS tracker on the land. And so that's like really at the forefront of the tracking, or underwater tracking, or getting like that really fine scale positioning of the fish. And that data, that stuff's amazing. Although, in contrast to the baited underwater stuff where you get lots of zeros in the data set, you get back a data set from this VPS system, and you're talking about 11 million detections of 60 to 100 fish over 6 months. So suddenly you get an 11 million rows in your spreadsheets, which it can't really handle that. But yeah, so you get a lot of data. Wow. A lot of data, well, much more than I expected, to be honest. So then we've been collaborating with a few other people. My supervisor from fisheries, he's got a few other species that are tagged around the marine park. He does some stuff on reef fish as well. I've just tagged a second species, so it's in the same genus as those blue spot flathead. I supervised an honours student, and we tagged some banjo sharks, so a type of flat shark that lives on the seafloor in the same area, and it's really common in this image. Well, it's really a ray, but call it a banjo shark sometimes. So we've got 3 species in there at the moment with tags in them. And the tags can last up to 3 years, the ones we have in, so you get quite a lot of data. So continuously having to go out and collect those receivers that I was talking about after, say, every 6 months, and then you download the data, put them back in the water, and repeat that until your batteries run out in the fish tags, basically. But that system, that whole array, is really quite a good system. I think it's becoming one of the better ones, and there's quite a few people that are starting to collaborate on it. There's people from Macquarie University in Sydney that are doing a whole lot of interesting stuff on Port Jackson's sharks in there, and their stuff is really interesting. I might try and tweet some of that later so you can have a look at that as well. Yeah, so the tracking side of things is something that I kind of fell into over the last 5 years, but it's become something that I'm really interested in. It's really interesting.
Yeah, I mean, that's some cool stuff. If you can pinpoint down to a couple meters where those fish are, that's really telling. You're not just guesstimating over these large swaths of ocean. You can actually say, we know they're here. And can you tell, like, you have, I'm assuming they give a code so you know which fish that is, so you can mark where a particular fish is traveling throughout that whole park then?
And you can start to work out that, okay, fish are probably not just sitting still and ambushing. But the big thing about fish on sand is because there's no obvious structure. So there's nothing that looks like the fish would hang around. So there doesn't seem like there'd be any reason for them to hang around. There's always been this kind of idea that fish on sand won't have a home range, or they won't show side attachment for very long because there's nothing really keeping them in any one position. But this tracking stuff that we're doing is actually showing, no, actually these fish look like they have a home range. Some of them for 300 days so far have just been using a tiny little couple of 100 meters by 100 meters areas and using it for that whole time. On sand, mostly to us, it looks really homogeneous. It just looks like there's nothing happening there. How do they know where they are? But obviously at some finer scale, they can tell the difference. But again, like in all things ecology, it's complicated, and they don't all do that. So some of them have done other things, like headed off to... I thought I had this really cool story, actually, that the fish are doing the opposite to what everyone does, and they have a home range. And that was after about 100 days of research. And I said, yeah, this is great. My story is easy to tell. It's interesting, and it's the opposite to what everyone expected. 200 days, yep, that was still the same story. And then about 250 days into tagging, when I was just pretty confident and was writing up, about a quarter of the fish turned up at Bondi Beach, which most people would have heard of. And that's about 180 kilometres north of where this bay is. And some of these fish are 30 centimetre long fish, so they're little fish that live on the bottom. For some reason, they just got up and left. And so that added a whole nother angle to the story. So there's something going on, maybe some sort of migration, or the adults are moving off, but we're still working on that one. But that's interesting. Maybe they like to go on holidays because Bondi Beach is pretty nice.
Yeah,
I
I
don't
always
know.
keep hearing about it. It's one of the beaches that you always hear about. When people talk about Australia, they always say Bondi Beach. So, yeah, maybe it's just
Yeah,
they had a holiday or on their way there. So,
yeah,
um, yeah, I think that's really interesting and telling too. Of we have, at least on the terrestrial systems, it's we kind of forget sometimes that species have these cycles, these annual cycles, or these, even multi-year cycles, and we have to make sure that as we're doing our studies, that sometimes it's so easy, especially when we're doing either master's or PhD projects, just to focus in on that one little area because we just want to be able to get that quick snapshot of what's happening, be able to write our paper and be able to graduate, and yay, we found something really cool, but it's only just a small little speck in their whole life journey, and there's so many different factors that could be impacting where they are and what they're doing. So
yeah. I definitely think you have hit the nail on the head there, and I guess it's a luxury in many cases to be able to do the long-term sort of research. And I mean, even I think there's 5 years of data that I have, including my honours. It's still a snapshot. You still want to have a long term. You want to have all different seasons, all different weather patterns. So lots of interesting results. But you also, like you say, you got to keep in mind that it is a snapshot.
but what's really
So
nice though is with the videos is that you can have people say 100 years from now, they actually can verify, okay, this is what it was you're seeing versus, when we pull up fish or species. I know when I go out and do surveys, I think I saw something, and I'll write down, what it is I believe I saw, but being able to have that photo, then I can actually truly have someone that verifies what it is that, that species that I identified, and you can have that person identify it 50, 100, 500 years from now, and I think it helps you create those more verified long-term data sets, because you kind of wonder sometimes with some of the data you have coming in from people, it's like, how accurate were they in those identifications of the species that they were seeing or the numbers that they were seeing? It's, when you're flying overhead on a plane trying to count the number of swans out there, it's kind of hard to do it if it's 1,000 swans. How accurate can you be? But if you take a picture of it, you can take your time and go through and count all of them. And same thing with your fish.
And then someone in 100 years can come back and use exactly the same technology, the same methodology, and do the same thing, which is actually really interesting. Because if I had the data from 100 years ago, we do have snapshots, so you have text them talking about various species that you see in this marine park, but they use different names that don't describe them in the same way that we do, so we're not entirely sure that they're even talking about the same species, which is kind of frustrating. If you had video, and you can just look back and say, oh, actually, they call it something different, but these are the same species, that would be really, really handy.
Right.
So,
I
really.
have the same issues as, okay, were they all talking about the same swans? Or they, what's going on? And so, it's, from, sometimes just region to region or time frame to time frame, there's so many different names of how people describe the same species, and you have to try to tease through that and figure it out. So it'd be nice to have that photo be like, yes, that truly is that.
Yeah, it would be handy. And there are moves afoot by various people. There's a researcher in Western Australia, Tim Langlois, to try and get all of this data from the bruvs onto like a central database and repository where at some point it all becomes open access as well, so people can get hold of this stuff as well, because there's thousands and thousands and thousands of hours of this footage sitting in storerooms all over the world, really. And it's a shame. I have more hard drives than you can imagine. I think I have like close to 80 terabytes of hard drives sitting in my office, and it's backed up in the fisheries office, and it's everywhere. I don't really have the capabilities to deal with that kind of huge file size and that much data, but it's probably the same all over the world. It'd be a shame if it all just gathered dust and eventually disappears. But we have the technology now that you could get it onto a big database and someone can use it in 5 years, in 10 years. So it will happen though. It will happen.
Yeah. Yeah, that's
Be
almost
very
kind of where
interesting
you
to
need to
see.
have it set up to do sort of a citizen science thing of where you can have people come in and start watching the videos and be able to catalog it all for you. Because, yeah, just one researcher, there's no way that you can watch all that. I know the few camera traps that I have around here that I'm playing with, just processing all of those pictures and videos, it's almost impossible. And I could only imagine a large scale project of any sort of like remote sensing like that. It just, I would never ever get through it at all. It's impossible, I think.
Yeah, there's definitely a lot of time spent sitting in front of a screen, yeah, analyzing videos. So yeah, there's, I don't know. There's got to be a way of it. Maybe it's citizen science. Yeah, it's definitely collaborating with people and getting a whole lot of people doing it, because it can be difficult to sit through hundreds of hours of video, even though a lot of it's really interesting. It can eventually get really tedious. Really, I like probably a lot of things where you're doing repetitive research can do that until you see the results and something in the hand. Then it gets exciting again. But I'd definitely recommend not individually going through that many videos. So, I mean, I have had help. He's doing a PhD now. I did supervise him doing honours, but he's done a lot of, for one chapter, he's done a lot of the videos in my project as well. So that was really, even just having that has been really handy.
Well, we're starting to kind of run out a little bit of time here, but I want to make sure that we fit in some of the Scicomm stuff, because so we were talking before the broadcast about how science communication has really helped us out, and you had a really good example of getting to, someone just saw some of your videos, I believe is what you said, and next thing you know, you're out on a ship doing research. So you want to kind of tell us a little bit about your
Yeah,
Scicomm story?
I know. We're talking about that. I think I was saying that I'm a latecomer to things like Twitter. I'm a part of a group called Fish Thinkers. That's a science communication group with another guy, Matt, and we do things on Instagram. And I was a late comer to Twitter, but I'm a big convert now. And yeah, so I think Twitter's great for collaborating, and that's the big thing that has made me realise that Twitter's a good thing. And the example that you're talking about is, so I'd filmed some saw sharks in my project, and then I'd been talking to Professor Jane Williamson. So she's a professor in Sydney, so not at my university. And I knew about her. I'd very much doubt she knew of me, other than through social media, basically. And yeah, so we talked about saw sharks, and she's done a whole lot more research in that area than me, but I'd filmed them. So we sort of touched base on doing something at some point, and then for me, it got put on the back burner. And then just out of the blue, Jane rang me and said, all right, we've got a boat going out in 2 weeks. We're going to put satellite tags on saw sharks, some of the first data that will ever be had on these sharks. And there's another guy, Yuan Morier, and another who's French. So I had Jane, who's from Sydney, me from the south coast of Australia, Yuan, who's French, and then Craig O'Connell, who's from the States. So he's a well-known shark researcher. And we came onto this research festival where a whole lot of projects were going on, and went out and did that. And the only reason that happened for me, from my angle, is from science communication, I guess, or from social media. So that, for me, is a good example of how useful it can be. And I mean, that was a great trip and really interesting research. So I think it's a good thing. I think, we were talking about that. I'm converted. So you don't have to convince me anymore that Twitter's a good thing, especially for two other scientists. I find it really useful, two other scientists. Whereas something like Instagram, we have a lot of interactions with recreational fishers and divers and people cleaning up marine debris. And that, I don't get that on Twitter. So they're both kind of complementary, I guess you could say. So
yeah, that's the thing with the different platforms out there. I find, like you said, that Twitter is much more of the scientist, where it's, all my colleagues, from, either in my field or other fields, that greater ecology, biology, conservation side of things. And then Instagram, I'm on there a lot, and I do find it definitely is more that mixture of science and non-science on there, of that particular audience. And So it's fun to see, people's responses that I'll get on Instagram versus what I'll get on Twitter. They're always just slightly different, and I always like seeing those different responses because I learn so much from them both ways, and the opportunities you get are really neat on both of those platforms.
Yeah, I think you're right. They're definitely different. Yeah, but seem like they're worth doing both. So.
Right. And it's just, it's a good, way for me too to learn, because as I'm, writing things about something on Instagram, I find that I'm learning about that particular species as I'm writing out and teaching people about it. And then same thing with on Twitter, I have people posting their journal articles or other research, and so I get that little quick access to that research that I might not have noticed otherwise through traditional channels.
Yeah, and you definitely, I mean, both have their pluses and minuses. Like I said, I think Twitter's a good thing, but it doesn't have the same appeal to youth, the youth side of things. So we have a lot of interactions with teenagers and young recreational fishers that we probably wouldn't ever interact with otherwise. And we've been invited to talk at like community talks and film opening as well, and things like that, and fish habitat workshops, and all these other things that it wouldn't have happened besides something like Instagram, which is kind of strange for me because I was also a little bit reluctant to get involved in that. I don't like revealing like my personal life. I like revealing my scientific life, which is a different thing. And so it took me a while to work out that you can do that. So, but yeah, I encourage people, scientists, to get on things like Instagram. I think it's pretty useful, and it's a different audience.
Yeah, I like how I feel like you kind of take away some of that mystery of science. It helps to those non-scientists out there. They actually get to see what is happening behind the science that they might not get to see otherwise, because they can't always come out on ship or go hiking through the woods as we go check to see what's happening. They have their lives that they're leading, and so they don't get to see it. All they usually see is maybe what gets shown on Discovery Channel, or like what happens to pop up on their local news real quick, and they may never read an actual journal article. So this way they actually can kind of see how science happens and maybe take a little bit away the mystery of it, and so that way they understand it and are maybe more likely to accept it when we say, hey, we need to fish less here because the fish populations have dropped and this is the reason why. And they'll be like, oh yeah, we saw that happening and understand. So
yeah, I think it can be very useful. And yeah, I wouldn't underestimate people on there either too. So you sometimes manage to get things like a paper to someone that otherwise would never have come across it, and they will go and read it. Surprisingly, scientific paper, no science background, they'll go and read it, and they'll come back to you with some pretty good questions that make you rethink how you're writing as well. Mm-hmm. So I find that pretty useful.
Yeah, like case in point, like I can just quickly throw up the paper that you did on the blue spotted flathead for everyone to see, and so anyone who watches this broadcast now knows about that paper, and if they're so inclined, they can go and read about it and find out a little bit more about your species and the result that you have from your study.
Yeah, so that sort of thing is good. But stay tuned for the next version of that paper too, because that's the real big one. Okay. lots of fish, and long term, that's going to be a cracker. Although I hope so.
Well, we'll have to have you back
if I can finish writing it. Yeah.
Well,
working on that now.
I think we're really starting to get way over time here, so we'll try to keep it short for everyone. So I'm going to thank Lachlan for being on. It was a great chat. I hope you had fun. Definitely feel free to come back anytime if you're doing field work or anything. We'll see if we can get that out for people to see.
No worries. Thank you very much for having me. And keep going with the show. I like watching it. It's really interesting.
Great.
All sorts of interesting research.
Well,
thanks again for having me.
Yeah, it was great having you on. So if people have questions that we didn't get to today and you want to ask afterwards, so if you're watching this broadcast on a replay, feel free to tweet Lachlan at Fish for Brains. You can tweet me at either the Scicomm Monday handle or at my personal handle of wildlifebiogal.com. Next week, we're going to have Janet Ng, known at Janet Ng Bio on her Twitter handle. She works with birds of prey, so if you like your hawks, all you ornithology folks out there, definitely tune in for that. So it should be fun. She does some really cool work up in Canada with the hawks up there. So with that, we're going to sign off and hope you guys have a great rest of your Monday. Make sure you go out, explore, do some science, and have some fun, and hopefully we'll see you next time.